If you were to commission an engineering firm to design a survival machine capable of withstanding the vacuum of space, traversing open oceans without propulsion, and manufacturing tensile structures stronger than steel, the result would likely appear distinctly artificial. They would construct a unit that utilizes high-pressure hydrostatics for locomotion rather than leverage-based musculature. They would integrate sensory arrays sensitive enough to detect the displacement of individual air molecules, and they would program it to extrude its own hunting infrastructure using liquid protein. We coexist with these creatures daily. Yet, when we strip away the familiarity, the common spider reveals itself as a physiological anomaly. These are not modern inventions, they are relics of the Silurian period, an ancient lineage established over 400 million years ago, predating the dinosaurs, the great conifer forests, and even the evolution of flowering plants. They are travelers from a time when the Earth's continents were barren rock. But deep analysis of their biomechanics compels us to ask a difficult question. How did an organism with a neural cluster the size of a poppy seed engineer solutions that modern material science is only just beginning to replicate? To understand this, we must view the spider not merely as an arthropod, but as a masterpiece of evolutionary engineering. We must first address a common classification error. To categorize a spider as an insect is a taxonomic inaccuracy. Spiders belong to the Chelicerata, a distinct subphylum that diverged from the ancestors of modern insects approximately 530 million years ago. While the insect lineage adapted for flight, the arachnid ancestors colonized the benthic and later terrestrial environments of the Paleozoic era. But the fossil record indicates that the transition to the modern spider was not a linear progression. Rather, it appears to have been a complex process of Mosaic evolution. For decades, paleobiologists hypothesized the existence of a transitional grade between the tailed arachnids of the past and modern weavers. Recent discoveries have validated this hypothesis, though they complicate the timeline. In the Carboniferous strata of France, dating back roughly 305 million years, we find Idmonarachne. It possessed the fused abdomen of a modern spider, having lost the ancestral telson or tail, yet it lacked spinnerets. It was a cursorial predator, a runner rather than an architect. In contrast, Burmese amber deposits from the mid-Cretaceous, roughly 100 million years ago, yield Chimerarachne. Despite being geologically younger, this species retained the primitive whip-like telson, yet possessed fully functional spinnerets. This data suggests that the spider body plan was not a singular invention, but a series of independent evolutionary experiments. Different combinations of sensory tails and silk production tools tested over millions of years before the modern Arachnidae order established dominance. A defining characteristic of the order is venom. Historically, it was hypothesized that arachnid venom evolved via the recruitment of digestive enzymes, essentially modified saliva. While this model holds true for many reptilian lineages, proteomic analysis suggests a different pathway for spiders. Current research points to a singular genetic divergence event approximately 375 million years ago. Researchers have identified an ancestral gene family, poetically termed Adi Shakti, which underwent an explosive duplication. This event generated a molecular scaffold known as the Inhibitory Cystine Knot. This structure acts as a hyperstable protein anchor. The spider's genome replicated this knot thousands of times, introducing slight mutations to the outer loops of the protein in each iteration.
This allowed for the development of a vast library of neurotoxins, each capable of targeting specific ion channels in prey nervous systems. It was not a digestive byproduct. It was the targeted development of biochemical munitions. There is, however, a notable deviation in the Chimerarachne family, specifically the genus Locomotion, known as the recluse spiders. Unlike the majority of spiders that rely on neurotoxic knots, Locomotion did recruit a metabolic enzyme, phospholipase D. This enzyme catalyzes a reaction known as transphosphatidylation. Upon injection, it shears the head groups off the phospholipid molecules that make up cell membranes. This reaction converts the victim's own cellular structure into signaling molecules that trigger rapid platelet aggregation and neutrophil activation. The result is dermonecrosis, the localized death of tissue. It represents a divergent and medically significant evolutionary strategy.
If venom provides the offensive capability, vision provides the guidance, particularly in the Salticidae, or jumping spiders. Lacking the cranial width required for the triangulation used in binocular stereopsis, these spiders have evolved a monocular solution utilizing a property of light often considered an optical defect, chromatic aberration. Lenses naturally refract wavelengths differently. Green light bends more sharply than red. The anterior median eyes of a jumping spider contain a retina with 4 distinct photoreceptor layers. The optics are tuned so that green light focuses precisely on the deepest layer, while appearing defocused or fuzzy on the layer directly above it. By processing the gradient of this defocus, the spider's neural ganglia can calculate distance with exceptional precision. It is, effectively, a biological rangefinder based on spectral physics. Their visual spectrum also exceeds human limits. While sensitivity to UVA light is common in arthropods, the species Phintella vittata utilizes the UVB spectrum for interspecific signaling. Males of this species reflect UVB wavelengths from specialized scales. Given that UVB is ionizing radiation capable of damaging DNA, this behavior is thought to represent an example of the handicap principle. By displaying in a spectrum that is environmentally hazardous, the male signals genetic robustness to potential mates, advertising that his biological repair mechanisms are superior. Perhaps the most distinct divergence from vertebrate physiology is the spider's method of locomotion. They function as hydraulic systems. While they possess flexor muscles to retract their limbs, they lack extensor muscles. Extension is achieved purely through fluid dynamics. To extend a leg, the spider contracts muscles in the cephalothorax, forcing hemolymph into the limbs and inflating them. In the Salticidae, internal pressure during peak activity during a ballistic jump has been measured at approximately 525 millimeters of Hg, roughly 70 kilopascals. To contextualize this, human systolic blood pressure averages 120 millimeters of Hg. The spider generates pressures exceeding 4 times that of a human heartbeat to achieve lift. However, this mechanism imposes a severe physiological limit. During peak activity, the hydrostatic pressure in the body cavity exceeds the pressure generated by the heart. Consequently, the heart valves are forced closed. This means that during a sprint or a jump, the spider effectively suffers temporary circulatory arrest. Its most explosive movements are powered by anaerobic metabolism, sustained only while the heart is stopped. Finally, we must reassess our understanding of arthropod cognition. The genus Portia, known for arachnophagy or spider eating, demonstrates problem-solving capabilities previously thought restricted to vertebrates.
Ethological studies utilizing expectancy violation protocols, similar to those used in developmental psychology, indicate that Portia possesses object permanence. If a prey item is obscured from view, the spider retains a mental representation of its location. Furthermore, these studies suggest a capacity for numerosity, or the ability to distinguish discrete quantities. When presented with distinct prey items that are subsequently hidden, Portia exhibits hesitation if the number revealed items does not match the initial observation. While their cognitive architecture appears limited to small integers, distinguishing 1 and 2 from many, the data implies a level of abstract processing that contradicts the model of arthropods as purely reflexive organisms. They exhibit numerical competency. They utilize sophisticated hydraulic propulsion. They perceive the world through spectral layers inaccessible to the human eye. Spiders are not merely remnant species. They are the Silurian architects. They have persisted through the Cretaceous-Paleogenic extinction event and the volcanism of the Permian. As we continue to decode their biological history, we find that they are not primitive, but rather highly derived organisms that have engineered survival strategies for 400 million years. The inquiry into their history is ongoing. If you wish to support the continued research and production of these documentaries, please consider joining our community. Thank you.